Good afternoon, everyone. I'm Micky, and I'll be walking you through the recent developments we have made towards multiplexed high-resolution imaging in mammalian nuclei. So to begin with, the 2-meter-long DNA, which all of us possess, gets wrapped around histones, gets reeled into loops by cohesin, gets organized into topologically associated domains, and then eventually gets packaged into this micron-sized nucleus. What might be appearing as a very packed, organized structure is actually quite dynamic. And the dynamicity of this DNA is sort of contributed by the various modifications on the tail of each histone component. And eventually what this results is, and then you have regions where the DNA is much more mobile, much more open, which we call as euchromatin. And then there are regions which are quite compact, quite silenced, the heterochromatin. And this balance is actually quite important because the cells need to maintain a sort of epigenetic homeostasis. Any variation in this resulting from lots of mutations or plethora of factors will eventually result in a state where the cell is not happy. It's undergoing some sort of genetic abnormality. So the question we wanted to ask is quite simple and straightforward. What is the nanoscale architecture of the epigenome in a mammalian nucleus? So how do we actually address this question? So we need two things. The first is we need a way to image things in high resolution so that we can look at individual proteins, because we are looking at several different modifications, several different proteins in a very small confined space. And the second thing is that because we are looking at so many different things, we need a way where we can actually multiplex them. We can image all of them at the same time. So let's look at how we can do that. And for that, DNA Paint is one of the best answers to this particular question that we have. And how does DNA Paint do high resolution imaging? So the idea is very simple. You have a target, and to the target, you can attach a small single-stranded DNA. And in your solution, you can have the complementary strands floating around carrying fluorophore molecules. At certain stochastic, you will have certain stochastic binding events where the imager and the docking strand will hybridize. You'll get a signal, and it will dehybridize because these are very short 7-base duplexes. So they won't permanently stay attached to them. And what you end up is an image which is just a set of blinking events, which we can simply reconstruct to obtain a high-resolution image. So that's basically, so now let's look at how DNA Paint works in cells. So what we are looking at here is a nuclear pore complex image, which is stained by the conventional immunofluorescence staining, where you have a primary antibody and a secondary antibody carrying the fluorophore. Now, if we were to replace the fluorophore with a DNA strand and then do DNA Paint, you can immediately see that we are now able to see individual nuclear NUP98 molecules in this nuclear pore complex. And the fact that we're able to see all these individual proteins together says that we have a resolution on the order of 20 to 30 nanometers. Now, once we have established that high resolution imaging can be done inside the nucleus, how do we do multiplexed imaging? And in that essence, DNA Paint is actually quite straightforward, that each of your target gets sort of barcoded with a unique DNA docking strand. So basically, if you're, so you can add P1 sequence, P1 star will only bind to targets carrying the P1 sequence. And so in that way, you can simply keep replacing the imager strands over several rounds, and you can simply overlay all of them, and then you can get a multiplex super resolved image. But the question is that how do we decide which is the best sequence? How do we make sure that the sequences which you are picking are actually correct? So the first thing we want to do is we want to get as many binding events as we can in the time in which we are recording that image. So if you are looking at this sequence, this is a 7 nucleotide sequence, which is TCC, TCC, T. The advantage what we have with this sequence is that this does not carry a complementary basis, so it will never form a hairpin. If we were to simply concatenate this sequence one after the other in, let's say, 5 different sequences, we could, in principle, increase the number of binding events by 5-fold. And that's the basic idea behind the speed sequences, where you can actually where we can actually acquire DNA Paint images at a much faster rate. In this case, at least, you have 5-fold more binding events in the same amount of time. And the other thing is that we should completely avoid complementary sequences at all costs, because even if it forms a hairpin, if this sequence were to form a hairpin, you would not get any binding event at all. So I would like to focus on the kind of, so what we can do is we can take individual blocks. So we can start with the 2 nucleotide blocks. You can have AG repeats or GAGA repeats. Similarly, we can take a 3 nucleotide block where you can have these blocks which are simply repeating and which can be concatenated one after the other. So 2 and 3 nucleotide blocks were already explored, were already published in 2020. We started working with the 4 nucleotide block where you have each block containing 4 bases which are repeating one after the other. And so these were the existing pool of sequences, the speed sequences which allow faster DNA paint imaging. We have come up with another set of 6 imagers with almost similar binding kinetics, which allow us to theoretically do 12 rounds of fast DNA paint imaging. And as a proof of concept, we took origamis carrying 12 unique docking strands, and we did simple exchange paint where each round of imaging, you're simply replacing the imager strand what you have, adding a different imager, and what you end up is a field of view where you have 12 different origami species, and each origami is shown with a different color because each is carrying a unique docking strand. So this sort of lays the groundwork that we are now able to image 12 different targets in the same field of view in 12 rounds of sequential imaging. So the next job we had to do was implement the same in cells. So we took the help of nanobodies, not secondary antibodies here. So we took nanobodies carrying a unique DNA strands. So you can have separate pools of nanobodies, each carrying a unique DNA strand. The idea is very simple. You take the primary antibody of your choice, pre-incubate it with a secondary antibody carrying a unique oligo. So in essence, you are basically barcoding this primary antibody with a unique DNA strand. And you can simply incubate them together, mix them, and then your final antibody cocktail has each primary antibody carrying a unique DNA strand. So that's job 1 done, that now we have labeled the targets. So now that we had all the antibodies coupled with their nanobodies carrying a barcoded docking strand, we started imaging several targets inside the nucleus, ranging from the transcription machinery, the splicing machinery, the euchromatin, the heterochromatin, and eventually ending up at the lamin. So we are covering factors which are predominantly located in the euchromatin and also factors which are predominantly located in the heterochromatin. And now I would like to skim through these images where we are actually looking at each target. So these are 9 rounds of DNA paint imaging of each target, and you can appreciate that. You can see lamin as a ring, you can see nuclear speckles as these sort of amorphous condensates, and then each of the nuclear targets. And now if we were to simply overlay them, we can end up with a 9-plex image where you are looking at all these 9 proteins in a single cell. So this is only stage 1 of the job done, that we got this image. Now, the other part is what can we do with this image? Because this is just a set of data points in a 2D plane. So now we started to do some very basic analysis where we are simply dividing these cells into grids of 25 nanometers. And now what we do is, if we take a simple grid over here and we count the number of localizations coming from each target in a grid, and now this will give us a pattern which will give us a sort of a global correlation between each target with respect to each other. And if we were to look at this cell, we can see that we are able to see expected patterns of co-localization where the active transcription machinery is well correlated with the nuclear speckles where splicing has to take place. The heterochromatin machinery is mostly uncorrelated with the euchromatin. So this sort of lays the groundwork that what we are seeing is actually true. And it's actually very much in line with what has already been reported, that there is some sort of segregation between the A and B compartments. Then we did something different. We inhibited transcription and we wanted to see what happens after that. So the immediate effect that we see is that the nuclear speckle machinery, which is sort of where active transcription is the mRNA is getting spliced, the speckles lose all contact with all the remaining histone marks. So that's the first sign that there is some amount of dramatic reorganization that is taking place in the chromatin environment within this nucleus. And so we started looking into the spatial correlation of the proteins with respect to each other. Because we had two defined structures. We had nuclear lamin, which forms the ring, and we had speckles, which appear as these amorphous objects. So we started with, we obtained the spatial correlation for each target for the lamin with all of those targets. So you can see that there are targets, if you're looking at from the point of view of nuclear speckles, the heterochromatin mark, for example, S3K and Me3, is more negatively correlated with your nuclear speckle, which shows that even with the spatial correlation maps, we are able to sort of obtain the spatial distribution of these proteins with respect to each other. But I would like to focus on these three targets for now. So what we are looking at is RNA-Pol 2 serine 2 phosphorylation, which is the elongating polymerase, RNA-Pol 2 serine 5 phosphorylation, which is the polymerase which is carrying the initiating mark, and then the euchromatin mark, which is K27EC. In both these cases, if you are looking at these three different proteins through the point of view of the nuclear speckle, there are a couple of things that we can see. The shape of the speckle is now more rounded compared to where you had a sort of amorphous distribution in untreated cells. Additionally, all these three targets have a higher degree of negative correlation with the speckles when compared to the untreated cell, where you have mostly highly positively correlated localizations. So this brings us to this particular image, where now we can see that you are also able to see the speckles carrying RNA polymerase 2 marks, and which is one of the reasons why you have a higher positive correlation. But the moment you inhibit transcription, the speckles barely carry any localizations from elongating RNA polymerase, resulting in this high negative correlation. Why is this happening? What's the basic fundamental process that's causing this? So this has actually been previously known, that actively transcribing regions are actively synthesizing RNA, and those RNAs are actually acting as the bridge between these nuclear speckle components, sort of pulling them apart. The moment you stop transcription from happening, the tether is gone, the RNA is no longer there, and as a result, the DNA sort of separates away from these protein components, and then the protein condensates are formally reverting back to their initial sort of circular state. So the key takeaway what we have from this sort of data is that we have now 6 additional speed DNA protein sequences, allowing us to image not just 6, but now 12 different targets. We have a way to perform fast multiplexed imaging in a mammalian nucleus, and now we have a comprehensive analysis pipeline which can make sense of all these multi localization data sets to obtain high density maps of spatial data. So I would like to end by thanking Mahipal's lab and also Abhinav, who has done a lot of work with the analysis for this. Thank you.
Thank you, Micky. Questions?
These are pixels. These are pixels.
And your speckled image, I mean, there seem to be some more structure in it. I mean, if you
just the speckle in the wild type cell?
Yeah.
Yes, those are sort of very heterogeneous structures. So there is not a uniform structure. Each speckle has its own shape. Some are long, some are like, but they are mostly amorphous. None of them are actually circular as we see in this particular case over here. So the moment you inhibit transcription, most of the speckles become these round donut shape, but
yeah, I see the difference, but
yes,
maybe there's some structure in the
some structure in these.
Yeah.
We haven't really analyzed the structure because these are, each cell has multiple speckles, each carrying a different shape. So we didn't really see a pattern there.
Hi, nice presentation. So I had like a few questions regarding the technique. So first is, how are you conjugating the nanobodies with the oligos?
The nanobodies have a single cysteine. So we use cysteine maleimide click chemistry to attach a functional linker. The linker carries DVCO. We have azide modified DNA. So it's a 2-stage click chemistry reaction.
And when you're like doing that co-localization experiment using grids, so like you're using some program software to
It's a custom written Python code.
Okay, okay.
So the modules are there. Just have to integrate it with your full pipeline.
And one last question. I know it's very hypothetical and very difficult to do, but is there any way we can integrate this technology into life cell? I know it's very difficult, but
I think this is something which comes up in every presentation we give. So the main issue here is that you're working with DNA strands. Getting these DNA images into life cell itself is tricky. Plus, life cells will be moving around, and we need something which is static to sort of localize this. So I don't think DNA paint at this point is capable of life cell imaging.
First of all, just want to say thank you for a fantastic talk, because I don't come from this space, no pun intended, but I still you're very clear about what you're trying to do. Second question was following up on the point was made earlier. I think, and you used the keyword already. You used the word heterogeneity. And I just wanted to point out the fact that I think by lumping all these measurements into a single global measure of either Pearson or spatial correlation, you might be missing out an opportunity to actually get into the patterning of these things in a much more resolved manner. So I would encourage you to perhaps think about ways to parse that a little bit more. If it's helpful, I am giving a talk tomorrow afternoon. I would love to get your perspective on using those kinds of techniques from spatial analysis for this kind of problem. I think it's a missed opportunity. So I just wanted to point out that you're onto something very important. You can follow up on that.
Sure, they look into it.
Thank you.
Question. So the nanoscale changes that you're seeing are a consequence of more also affecting
Yes.
large-scale chromatin
Yes.
organization, and therefore you're still seeing these nanoscale
Yes.
changes. Can you actually look at perturbations that likely not affect global architecture locally? Can you pick those up?
We are actually thinking about some of those experiments where we can actually do some site-directed stuff in the cells, but that's still a long way ahead.
Micky.
Yeah, that was really cool work. Can you get information on stoichiometries from your method?
From this data, no, because we are using nanobodies, so we have some labeling artifacts. But the next round of data, what we are getting is we are directly conjugating primary antibodies. So that should give us a better chance of at least measuring some crude amount of stoichiometry from there. It's again limited by how efficient your antibodies are again. So that's the major hurdle we have over here.
You know what approximate labeling efficiency you're achieving?
No, not unlikely, but the best case, what I've heard from people is 40 to 50%, the best case, not more than that.